Imagine a world where male and female gametes can't move on their own. In flowering plants, that's exactly the case. Both the male and female gametes are non-motile, unlike in animals where gametes often travel to meet each other. So, how do they ever meet? That's where pollination steps in, a crucial event that bridges the gap between pollen grains and the stigma of a flower. But it's not just a random transfer, it's a highly adapted process, fine-tuned over millions of years of evolution. Pollination is nature's way of making sure that even without movement, life finds a way to begin. Pollination doesn't follow a single route, it varies depending on where the pollen comes from. There are three main types of pollination, classified based on the source of pollen: autogamy, the simplest of all. Pollen is transferred within the same flower. No movement, no travel, just a quiet self-transfer. Geitonogamy, a bit more social. Here, pollen moves between different flowers of the same plant. It may look like cross-pollination, but genetically, it's still a form of self-pollination. Xenogamy, this is where diversity blossoms. Pollen travels from one plant to another as long as they're of the same species. This is true cross-pollination, and it's the main driver of genetic variation in plant populations. Among the three, xenogamy plays the biggest role in strengthening plant traits and boosting adaptability over generations. In short, how pollen moves shapes how plants evolve and thrive. Pollination doesn't happen by chance, it depends on the help of pollination agents, both abiotic and biotic, each perfectly adapted for the task. Let's start with the abiotic agents, wind and water. Wind pollination, or ranomophily, is common in grasses. The pollen? Lightweight and non-sticky, made to drift freely through the air. The flowers? They have exposed stamens and feathery stigmas, specially designed to catch even the faintest breeze. Then there's water pollination, or hydrophily. It's rare, but truly elegant. In plants like Vallisneria, pollen travels on the surface of water, gliding gently toward the female flower. Now let's meet the real showstoppers, the biotic agents. These include insects, birds, bats, and surprisingly, even lizards. Flowers pollinated by animals are often brightly colored, scented, and rich in nectar, a perfect reward system for their visitors. And some flowers go even further, offering shelter or even a place to lay eggs, just to keep their pollinators coming back. It's a finely tuned relationship, nature's perfect alliance between plant and pollinator. Most flowering plants produce hermaphrodite flowers containing both male and female reproductive organs in one place. Sounds efficient, right? But there's a catch. Continued self-pollination can lead to inbreeding depression, reducing genetic diversity and weakening plant vigor over time. To prevent this, plants have evolved a set of clever strategies called outbreeding devices, natural mechanisms to encourage cross-pollination and ensure healthy, varied offspring. One such strategy is dichogamy, a form of temporal separation. Here, the pollen is released either before or after the stigma becomes receptive, making self-fertilization nearly impossible. Another is hercogamy, a physical separation. The anther and stigma are positioned in such a way that pollen can't easily reach its own stigma. Then comes self-incompatibility, a genetic safeguard. Even if self-pollen lands on the stigma, the plant actively blocks its growth, preventing fertilization from taking place. Some plants produce unisexual flowers. In monoecious species like castor or madge, male and female flowers grow on the same plant, preventing autogamy, though geitonogamy may still occur. But in dioecious species like papaya, male and female flowers grow on separate plants entirely, eliminating both autogamy and geitonogamy. Each of these adaptations, timing, structure, genetics, and flower distribution is nature's way of saying variation matters. Pollination may deliver the pollen to the stigma, but not every pollen grain is welcome. What happens next is a selective interaction called the Pollen-Pistil Interaction. As the pollen lands, the pistil evaluates it. Is it from the right species? Is it genetically compatible? This isn't guesswork, it's a chemical conversation between the pollen and the pistil. If the pollen is compatible, it germinates on the stigma and forms a pollen tube. This tube begins its journey through the style, carrying to male gametes toward the ovule. In some plants, the generative cell divides during this journey. In others, the two gametes are already formed before the pollen is released. As the tube approaches the ovule, a guiding structure called the filiform apparatus directs it into a synergid, perfectly positioned for fertilization. But if the pollen is incompatible, for example, self-pollen in a self-incompatible species, the pistil blocks the germination or stops the pollen tube's growth, preventing fertilization altogether. This interaction is nature's own quality control, a powerful checkpoint that ensures only the right pollen reaches the ovule. And in plant breeding, it's a valuable tool, helping us create hybrids by carefully selecting which pollen makes it all the way. In the world of plant breeding, precision matters. To create hybrid varieties, scientists use a technique called artificial hybridization, a process of controlled cross-pollination. It all begins with emasculation, gently removing the anthers from a flower before they can release pollen. This step prevents self-pollination. Next, the flower is bagged, covered with a protective layer to shield it from unwanted pollen in the environment. When the stigma becomes receptive, breeders carefully apply pollen from a selected male parent. This ensures that the cross is intentional and precisely controlled. The result? Genetically superior crops with desired traits like disease resistance, higher yields, and improved nutrition. Artificial hybridization is not just a method, it's a powerful tool driving the future of sustainable agriculture. As we wrap up this journey through pollination, let's connect the dots. Pollination isn't random, it's a strategic process that brings together male and female gametes in flowering plants. We explored the three types: autogamy, dichonogamy, and xenogamy, each with its own role in reproduction. We saw how wind, water, and animals like bees and birds act as pollination agents. To avoid inbreeding, plants use outbreeding strategies like dichogamy, halcogamy, self-incompatibility, and unisexuality. Then came the heart of the process, pollen-pistil interaction, where the pistil becomes a gatekeeper, accepting or rejecting pollen based on molecular recognition. And finally, we saw how humans have harnessed this knowledge through emasculation and bagging to engineer better, more resilient crops. Pollination is more than a transfer, it's a symphony of timing, recognition, and adaptation, ensuring not just reproduction, but survival and success across generations.